So we're all pretty familiar with bronchiolitis as this sort of commonest reason that a child ends up in PICU under a year of age for an unplanned admission. In fact, all of the variation in unplanned admissions to PICU is created essentially in the winter by bronchiolitis under a year of age. Anyway, the predominant age for children admitted to PICU is under a year of age anyway. The median is well under 3. So here's that unplanned admission each winter, and you can see each winter we have a surge of respiratory admissions that in the UK tends to coincide with the coldest point of the year. So it's often around Christmas or the end of December. And you can see that there's a predictable peak, but an unpredictable intensity. But it's always been there. And year on year, if you look at unplanned admissions for non-acute respiratory failure, non-bronchiolitis, they're pretty flat right through the year, but the surge is the winter with acute respiratory failure. So the predominant viruses that I might talk about, I mean, coronavirus really doesn't cause bronchiolitis, but I thought I'd talk about some of these viruses as we go along. We might get to coronavirus, we might get too bored by that stage. So where was I? Yeah, there's the unplanned admissions under a year of age and the difference between acute respiratory failure and non-acute respiratory failure. So one way we can look at viruses is by stratifying them into whether they are those that use reverse transcriptase, whether they're DNA viruses, whether they're RNA viruses, whether they are single-stranded or double-stranded, and then for RNA viruses, whether they are in a positive sense of RNA or a negative sense of RNA. And these can be classes. So we class 1, 2, 3, 4. This is just how arbitrarily it's been used for many years. And if we then look at that a little bit more blown up, then you can see which viruses belong roughly to which families. So if we go down the DNA route, we can see where these big clunky herpes viruses and pox viruses and wart viruses are. And we can then look across the RNA viruses and we can see which ones are single-stranded, because the double-stranded ones don't appear to be an issue for humans. But the single-stranded RNA could be in a positive sense of RNA or a complementary negative sense of RNA, single strand. And if we then look at whether they are enveloped or unenveloped, we can see that we can subcategorize them again. So we've got things like hepatitis A, rhinovirus, enterovirus in unenveloped and positive sense single-stranded RNA viruses that are not associated with reverse transcriptase. And then we've got RSV, which is a single-stranded negative sense RNA virus, part of what are known as the mononegavirales. So there you go, it's nega as in negative sense, mono as in single-stranded viruses. Yeah, so we can subcategorize them. You can see roughly where the viruses that we're going to be talking about fit. So human metapneumovirus and RSV are in the mononegavirales, where measles is too, and then the other predominant group, the rhinoviruses in particular, which look very like enteroviruses. That's why our set of primers on the PCR often comes up with a positive virus for enterovirus. And they come into the group that are known as pico-RNA viruses, picornaviruses, pico being very small RNA viruses. So often it comes up with a positive picornavirus result on the MPA, and then it says enterovirus, but actually they then isolate it rhinovirus, and that's because of the primer set that they use to amplify it. So if we look at who's being admitted, it's a lot of children. So this is the whole of PICU in the UK over a period of 4 years. And you can see that there are about 26,500 admissions under a year of age. And of these, about a half are actually unplanned, and a big chunk are unplanned with acute respiratory failure. So it's about 1.8 infants per 1,000 per year are admitted with bronchiolitis. It's quite a substantial number of kids. If you look at the data in a little bit more sort of detail, you can see that a number of read codes could come into the group of acute respiratory failure. So there's bronchiolitis, acute bronchiolitis due to RSV. There are a number of ways that they could be coded that would cross and overlap. You know, we've got acute viral bronchiolitis in there as well, acute lower respiratory tract infection, other. So a lot of these things do group together. And for the purposes of this paper, what we wanted to understand was whether there were any factors like ethnicity or deprivation or prematurity that had a role to play. And what we can see is that being admitted to PICU bronchiolitis is substantially more common if you're from a Southeast Asian origin versus a non-South Asian origin in the UK. And mortality is higher in that group too, although the mortality overall is very low. And what we can see as well is that deprivation in terms of the Townsend score is a mark of deprivation. So you're much more likely to be admitted with bronchiolitis if you come from a deprived postcode than if you're in a more affluent postcode. Overall, the length of stay is pretty predictably 5 days for most children. And so although mortality was slightly increased in deprivation and admissions were increased in deprivation, and mortality and admissions were increased in South Asian infants, there's no overall effect on the length of stay for these children. Prematurity is an interesting one. So there are about 7.5% of the UK population are born preterm, quite a substantial number. And if we look at how they do on PICU, overall, there is a much higher chance you've been born preterm of ending up with bronchiolitis on PICU, almost 40%, even though the UK prevalence of prematurity is only 7.6%. And they have an overall longer stay on PICU, but the overall mortality is actually very similar. So they actually survive bronchiolitis, and we couldn't detect it with the power of this study any statistical difference in terms of their survival. So I think a lot of people are fairly bleak about kids who've been born preterm with bronchiolitis, but I think we should be fairly optimistic actually.
So in this study, did you also kind of divide them into immaturity, like late preterm, early preterm,
no,
extreme preterm?
Just anybody born before 37 weeks. So this is electron micrograph of RSV, and RSV is, well, I think it's a very interesting virus as an archetype. It's so fragile. It's only a single sense of negative stranded RNA. I mean, UV light breaks that down, so it's sensitive to sunlight. And yet it manages to infect the entire population of the planet by the time we're 3 years old. So it's a very, very fragile virus, but somehow it manages to get us all. It's got a few surface proteins, 3 predominant surface proteins, an attachment protein, a small hydrophobic protein, and a fusion protein. And the other proteins are internal. And in particular, there are very highly conserved proteins like the nuclear protein and phosphoprotein that aren't really on show to the immune system. So the fusion protein and the attachment protein, the G protein, they show a lot of variation between different virus strains because they're under continuous antigenic pressure from our immune system and antibodies. So they're very highly variable proteins. But the ones within the envelope are within the lipid bilayer. They are highly conserved proteins, the polymerase, nuclear protein, and phosphoprotein. And if we look at those highly conserved proteins, they are quite distinct. So if we look at other viruses, so you remember I was saying that within the mononegavirales, things like measles. But if you look at paramyxoviruses, which include the which are included in the mononegavirales group. Actually, although RSV looks like measles macroscopically or microscopically, there's the polymerase and the nuclear proteins actually show much closer homology to filoviruses. So they probably had a common ancestor several thousand years ago that was closer to Ebola than the innocent virus that we take it for today. So it's actually interesting how probably it's adapted to human beings over a long period of time. And obviously, if you kill human beings, it's very difficult to pass yourself on and replicate. So it's to your advantage to become less and less pathogenic as time passes. It's spread by things that we all do. So we all rub our eyes and we all pick our noses. And the virus has established that as its environmental niche. So it likes to live in our noses. And it doesn't actually infect oropharyngeal mucosa. So if you swab their mouth, there's no RSV there. It only likes respiratory epithelium associated with the nose and the throat and the lungs. This is a RSV budding from a cell surface. And it is everywhere. By the time we get to 3 years of age, everybody on the planet has had RSV. It doesn't matter if you go to Arizona or if you go to Alaska. By the time you're 3, the seroprevalence to RSV is pretty close to 100%. It's very good. So your first infection is going to be in the first few years of your life. And interestingly, the earlier you have it, the more likely you are to have recurrent symptoms and the more likely you are to have asthma or atopy later on. Initially presenting often as eczema, but then progressing to full asthma. The question is that nature or is that genetics or environment? It appears from some work done by a lady called Nelly Siggers, who is a Swedish scientist, that there appears to be a very strong environmental association with early inflammation. So if you put a child onto cardiopulmonary bypass to have a cardiac operation, their risk of being sensitized through that process to environmental antigens and going on to have eczema or other forms of atopy is higher than other children. And children who are admitted with bronchiolitis and receive intensive care have a far higher risk of being sensitized to environmental antigens. And this is made worse by factors such as pollution and cigarette smoke exposure. If we look at when RSV infections occur, there's quite a lot of seasonal variation depending on the climate of the country that you're in. So in the UK, which is a sort of fairly temperate climate, we have the seasons that are fairly distinct. And in countries like ours, we have peaks during the coldest point of the year. If we look at other more tropical or subtropical environments, such as Brazil or Vietnam, you can see that there's a much closer association to humidity relative to temperature. So we actually have an inverse association with humidity, whereas Vietnam has a clear association with humidity. So it's not really clear what it is about the climate that makes infants susceptible to RSV. But it is interesting how there's variation around the world.
Recently, in the last 2-3 months, in Journal Pediatric Wards, there has been kind of lots of RSVs. I don't know, like usually it was always like November, December, January, February. But in the last 2-3 months, whenever I speak to my friends, they are like, they are all RSVs.
I think so far this year we've had an earlier onset of RSV bronchiolitis. We haven't seen the surge that we would normally see in the winter yet. The question is that going to be something down the line that we need to take account of? I guess, time will tell. Ask me again next year. So, hi.
Hello, is it a good time?
I'm giving a lecture.
Oh, I'm sorry.
No, it's okay.
Is there anyone else around? I just got a very quick question about little Harriet and her tracking.
Ask away. What's the question?
Basically, we're trying to get. Mum's just brought up the the idea that Graham, her partner, isn't tracking tube change. He has never done a tube change.
Yeah.
And ideally, she'd like to go home back with a bivonium that she had before.
Yes.
So my question to you is, would you be happy if I supervised the tube change back to a bivona?
Yeah, totally.
Is that all right?
Yeah,
Thank you.
absolutely. I think Mum is quite happy that. Jolly good. Right. So are there any experimental parallels? You know, I was talking about Nellie Sigger's work about allergic sensitization. Well, we can do this in mice. So if you look at mice and you infect them with acute respiratory viruses like RSV and flu, during the acute infection, they look unwell, they lose weight, and then they recover and regain their weight. If we do a three-way experiment where we mock infect them, we give them RSV or we give them flu, and then we expose them to an allergen, and for this we used egg white, we used ovalbumin. You can see that both RSV and flu can stimulate the production of the mouse equivalent of IgE, which is IgG1. So if you measure their plasma afterwards, they develop an allergic response to egg white if you expose it during the context of an acute viral illness. If you expose it after or before, they don't develop it. So there's something about inflammation in this context that allows sensitization to take place. There's a lot of immunology there that I might actually leave, but one thing I should point out, that one of the questions that often comes up is why don't we have a vaccine to RSV? It's such a common illness. And the reason is that back in the 60s, there was a rush of vaccines produced to a number of pathogens. After Salk and Sabin's work in polio vaccination, which was absolutely groundbreaking, formal inactivation of a virus was used for immunization as a strategy. And there were several trials simultaneously of formerly inactivated RSV in this context. And what happened was the children presented got really good antibody responses. They sat back and waited for the winter. But unfortunately, what happened was they got an increased illness. So they got vaccine enhanced bronchiolitis. And a number of children actually, so the admission rate went from 5% to about 80%. And a number of children actually died of bronchiolitis who wouldn't have been expected to. Now, this is many years ago, but it has created a lot of that concern. Hi,
so
Chris.
Sorry, I've just got NNM ringing about Harriet.
Could you? Okay, so we've had, quite a number of years, 50 years plus since then, but we still haven't got a reliable RSV vaccine.
Yes, help me. He is not in this week. We're trying to figure out if we can get hold of someone else who can do it.
Quite right, quite right. The other feature of this is there are no known natural reservoirs for RSV in the animal world, in the animal population. The only animal that consistently been found to be infected with RSV is humans. And the question is, where does it go? Because it seems to reappear every winter promptly. And one of the questions is whether, like other viruses, it might establish some sort of latency somewhere and then re-emerge in order to spread to new hosts. Certainly in the mouse population, if you can infect them with, you have to infect them with a lot more virus than you do for humans. If you can infect them, you can actually detect it. If you mush up their lungs and PCR it, you can see that there is some persistence even 90 days later in the lung. So there may be the ability of, it hasn't been proven, but the ability in humans is possible that there could be latency there too. Other features of RSV, which are common actually in some other virus infections, bronchiolitis as well, is the phenomenon of lymphopenia. So lymphopenia in RSV bronchiolitis appears to be associated with severity of illness. So as a surrogate for severity in this study, I've looked at the ward and PICU as separate areas. And what you can see is the overall lymphocyte count declines the sicker you are. So your lowest lymphocyte count in PICU compared to milder infections on the ward, compared to those who had RSV but did not get admitted. You also have a subtle but statistically significant neutrophilia associated with bronchiolitis. And if you look on the lymphocytes themselves, there is upregulation of CD95, which is also known as FAS, which is one of the pathways through which apoptosis takes place. So it appears to be a lymphopenia related to actual lymphocyte apoptosis as part of the inflammatory response to RSV. Younger infants also have more significant lymphopenia, and they also tend to be sicker than older infants. Why is this potentially interesting? Well, we see lymphopenia also in septicemia and in mouse models of septicemia. If you inhibit some aspects of apoptosis, you can promote survival of individual mice compared to those who do not have apoptosis inhibited. So there are potential therapeutic implications down the line of both in sepsis and in bronchiolitis of manipulating immune response. Human metapneumovirus, which you've probably come across a lot now, and probably take it entirely for granted, but actually it was discovered this century in RSV negative bronchiolitis patients in Holland. So this group discovered and identified another single sense RNA negative strand virus that looks a lot like RSV in many ways, and belongs to the same pneumovirus family, called human metapneumovirus. And this also has very widespread infection, almost as far as RSV across the population, and is often associated with severe bronchiolitis, although there are fewer admissions with human metapneumovirus than there are with RSV. So this is how the family looks now. We've got pneumovirinae here and paramyxovirinae here. So in the paramyxo viruses, we've got morbilliviruses, the measles viruses, and we've got rubella on this side, and we've got some of these respiratory viruses, parainfluenza virus. On this side, we've got RSV in the pneumovirinae, RSV, and human metapneumoviruses as a separate offshoot of the mononegavirales. So human metapneumovirus, very similar structure. It's got a short hydrophobic surface molecule, just like RSV, a glycoprotein, highly variable, just like RSV, and a fusion protein. And it's got an RNA complex with matrix proteins, a polymerase, and a phosphoprotein. There are antibodies that have been identified in human serum for human metapneumovirus going back to stores from the 1950s, which is about as far back as we can go. But probably it diverged about only 200 or 300 years ago from avian viruses. We haven't yet worked out how it binds to human beings to enter the body. Okay, I thought I'd just do if people are interested, I thought I'd do a little bit of a refresh about a bit of physiology.
That would be good.
Would that be helpful in terms of understanding why children might be particularly at risk? So if we remember that in the pleural space, there's an overall negative intrapleural pressure that's created by the combination of ribs trying to expand out and being pulled in by lung trying to elastic recoil. And so in an adult, our lungs are held in a position where the airways are held open in the perfect position to have the minimum resistance to breathing at tidal breathing. And this perfect anatomy creates that environment. But in children, the ribs are not fully formed. And so there's still a lot of cartilaginous soft rib cage. And so this outward pressure is less powerful. So what we see when respiratory effort increases is deformation of the rib cage with intercostal and subcostal recession, as the lungs can't be held in a position where they are ideally open. and therefore at low resistance. And this impacts on our hysteresis curve and compliance. So if this is our normal inspiration and our normal expiration, the difference between these two sides is known as hysteresis, and is a property of the way surfactant expands and then is compressed, because surfactant molecules obviously repel one another, and at lower lung volumes and small alveolar size, we get heaping up into several layers of surfactant, which then spreads out very nicely on inflation. But the popping up process is something that requires a little bit more energy. Therefore, it is slower to happen on deflation. This hysteresis keeps airways open. And we can see also that the pressure against the volume of the lungs is very important. So if the slope of the curve is the compliance of the lungs at that point, normally in an adult chest, our lungs are kept in the position that is most compliant by this interaction on the previous page. So the fact that the ribs are holding their chest at a certain volume and the elastic recoil is balancing that out is holding them in this range of tidal volume, where the change in pressure for a given change in volume is smallest. So a small change in pressure creates a big change in volume. So it's a very steep and optimal place to breathe. You don't have to generate huge pressure changes in order to get a volume change in tidal volume. However, if you overinflate the lung, you reach a point where any change in pressure has to be quite substantial to generate any further change in volume. And that's created by reaching the full expansion of the rib cage, essentially. And at lower volumes, if you're breathing at lower volumes, essentially it's the same. Little airways start to close, or you get collapse of alveoli, and opening them up again takes more pressure. So at a less optimal point here at low volumes, your compliance is reduced. Another factor, so you can imagine that if you have a change in volume related to inflammation and a change in compliance related to inflamed, stiff lungs, you can imagine, or a change in elastic recoil created by inflamed, swollen lungs, that the balance point will be different for those individuals. And that's what's happening in bronchiolitis. There's also the issue of oxygen diffusion into water. So actually, oxygen diffusion into water means that the amount of oxygen in the water is at a lower level in terms of the millimoles per liter than it is in air, because it doesn't dissolve very well in water. Oxygen does not dissolve well in water. Carbon dioxide does dissolve well in water. And the other issue is that the thing that creates the gradient, which allows the oxygen to dissolve in the water, is created by the difference in partial pressure of oxygen in the atmosphere with that in the water. So it's driving them to an equilibrium point here. And at equilibrium, there's a lot less oxygen in water than there is in air. Does that make sense? What this means is, I should also say there's this factor as well. There's also this concept of diffusing capacity. So the transfer of oxygen from the air in the alveoli into the water, which is the plasma, is also affected by the membrane of the alveolus and any inflammation that's associated with the alveolar pulmonary vascular interface. So if you've got inflammation going on in that bit of or edema or swelling, you've got an increase in the gap that allows oxygen to diffuse into the water. It's got to diffuse through swollen, inflamed tissue, and that will slow down the transmission of oxygen into the plasma. Now, normally, if this is the entire transit time through the lung, which is less than a second, normally diffusion occurs in less than a quarter of a second. So less than a quarter of the time it takes for the red cell to move from one side of the pulmonary circuit to the other side of the lungs. It's already it's already had oxygen added to it. If you have a change or a limitation in the diffusion of oxygen across from the alveolus caused by edema, that can be slowed so that it takes actually the whole time of the transit to absorb or diffuse across the inflamed alveolar wall. And in severe inflammation, you may not get complete diffusion in time to get oxygenation in the blood. Even taking that into account, in normal situations, there is, as you know, a drop-off in the PO2 in different parts of the circulation. So remember, we were talking about air, which is dry, conducting airways, which may well have a bit of carbon dioxide in them, falling into alveolar gas, by which stage there's also been humidification. So there's water in this alveolar gas that's taking up some of the space. So although the overall partial pressure is 760 millimeters of mercury, the same as it is at sea level outside the lung, there's going to be a fall off as things are added, CO2 and humidity being major factors. So that alveolar gas has a lower PO2 anyway, with which to drive this equilibrium compared to the air that we're breathing. The other factor that we have to consider, apart from diffusion capacity and the partial pressure of oxygen, is a concept known as closing volume. So in normal tidal breathing here, airways are maintained open by that relationship between the skeleton that I showed you before, the chest, and the elastic recoil of the lung, and the negative pressure in between the two in the pleural space. So they're kept in a position where airways do not close. But if you were to start to breathe out beyond tidal breathing, you can, in some individuals, reach a volume at which, if you look to the right of the curve, you've got a little bit of closure or narrowing of smaller airways. Now, in general, adults can't breathe, healthy, fit adults, young adults, can't breathe out to the point at which they reach closing volume, because their expiratory reserve volume won't allow them to do that. However, in infants, where they've got this pliable chest wall, infants can, particularly newborns, can actually breathe out to the point at which the airways start to close like this in normal circumstances. The way that they avoid that is by dynamically recruiting FRC by taking the next breath before they reach closing volume. So they've got stretch sensors in the lungs that detect that they're about to receive, they're about to reach closing volume, and they take the next breath. Clearly, if you have inflammation there, you might reach closing volume sooner, even in tidal breathing here. And so what does the infant do? It breathes more quickly. So tachypnea is the first feature that we're looking for. Now, you can only breathe out up to a certain maximum rate, and that depends on elastic recoil and the distance over which the lung has to move. And children have great elastic recoil generally, but and the distances that their lungs have to move are smaller, but there are still limits to the respiratory rate that they can generate, and that's somewhere around 100 or 110. So an infant can actually raise its respiratory rate up to about 100, 110, maybe 120 in some small kids. And at that point, they can't dynamically recruit FRC, and they will be reaching closing volumes. So what they do at that stage, which is a reflex, is they partially close their glottis and they breathe against a partially closed glottis, and they make a grunt. So they go, So in the absence of pain, that grunting implies alveolar disease and that their stretch receptors are reaching closing volume. So it's a very clever reflex. Now, we might take that into account in terms of the way that we ventilate these children. So what we're essentially providing is a back pressure against which we avoid this closure using PEEP, for example. And we also need to consider how quickly their elastic recoil allows their lungs to contract. And in obstructed bronchiolitis, like this, clearly gas movement may be obstructed. We then have to consider lengthening the expiratory time, and that's often at the expense of the rate, so the rate has to come down. So compared to a newborn that we can often ventilate at very rapid rates, 60 per minute, you can't do that to a kid with bronchiolitis because of those two factors.
Right.
Right, okay. Other things, coronaviruses. Very quickly, coronavirus is rare cause of bronchiolitis. But if we look at the history of coronaviruses, these are from a different family. So these are a positive sense RNA virus. They are enveloped. They tend to cause illnesses in the winter and early spring, and they've been around for all of our lives, clinical lives. And they're across the globe, they are ubiquitous. Interestingly, they come from the same virus family as dengue and Zika, and they affect farm animals and pets. It's striking that the first coronaviruses were discovered in domesticated chickens. That there used to be epidemics of coronavirus that killed whole barns of chickens, and that from this discovery, coronavirus was isolated. It was first virologically identified in humans in the 1960s as a cold virus. It's very interesting that the most recent common ancestor is perhaps only 8,000 years ago for this virus. The longest, if you look at the slow evolution of the virus and you track it back from previous isolates, probably only goes back for about 8,000 years as a class of viruses, not that long. The structure, however, is, well, it's all familiar to us now. We've got a spike protein on the surface, an M protein, and a hemagglutinin esterase dimer. There's an envelope, and then there are internal proteins, which are nuclear protein and the RNA positive sense. So it's an encapsulated envelope virus. So how did we start to get worried about them? Well, you'll remember SARS, and this guy was an absolute hero. He was the one who prevented SARS from really taking a foothold by recognizing that something was going on. So he's a World Health Organization ID expert, born in Italy, Carlo Urbani, who worked on hookworm and schistosomiasis. But he was invited to give advice. He was visiting at a conference, Hanoi, and he was asked by a colleague to come and look at a patient called Johnny Chen, who was an American businessman who'd been admitted to hospital and become critically ill with an acute respiratory illness that looked like influenza. But he was, with his World Health Organization ID hat on, he was able to recognize this was a new phenomenon associated with the coronavirus. And he alerted the Vietnamese government and the World Health Organization. Sadly, he caught the virus, the coronavirus, from Johnny Chen, and was hospitalized and subsequently died, but not before he had warned the world that SARS was coming in March 2003. So he is he's an ID hero, and he should be somebody remembered. It was despite people scrambling around the world to prevent it happening, it was a significant outbreak. Nothing to the current coronavirus outbreak, but there were about 8,000 individuals who developed SARS, and the mortality was substantial, about 10%. So it was higher than coronavirus, and it particularly affected healthcare workers. So there were a lot of healthcare workers who were killed by SARS. It was noted that the cellular receptor, like the coronavirus that we're seeing now, COVID-19, was angiotensin converting enzyme 2. Now, this is not angiotensin converting enzyme, the one that we normally think about. This is not the one that changes angiotensin 1 to angiotensin 2. In fact, this is an enzyme present particularly in the lung that converts angiotensin 2 back to angiotensin 1, which has very different vasoconstricting and vasodilating properties. So there's a lot of mix-up about that in the literature, but you know now that that's the case. So after SARS disappeared, we had another coronavirus outbreak in 2013 called MERS, that arose in the Middle East and caused respiratory symptoms. So it was called MERS. There were about 700 cases reported, mainly in Saudi Arabia, with 300 deaths. So this is a very high death rate of the cases that were identified. It was already known to be similar to strains in Pipistrellus bats, and it was particularly associated with acute renal failure in these individuals. It was quite a long incubation, which is why the incubation time, the isolation time, is so long for coronavirus, and it appears there is some commonality with coronavirus incubation. Interestingly, Pipistrellus has a peri-Mediterranean distribution, a little bit down in Madagascar. And then we come to COVID-19. So why are people worrying about vaccination of children with COVID-19? Well, there have been a lot of studies on viruses from the same Baltimore group, dengue and Zika, which cause inflammatory syndromes, multi-inflammatory syndromes. And there appears to be antibody-associated enhancement for those two infections. So the worry is whether the inflammatory syndrome, the multi-system inflammatory syndrome that we're seeing in children, which appears to be antibody-associated with myocarditis and skin rashes and diarrhea, whether this is actually antibody-enhanced illness or not is an open question. So you can see that you get rashes with Zika and dengue in the same way that you do, with very similar rashes to what you get with the multi-inflammatory syndrome. So in summary, the annual variation of unplanned respiratory admissions is basically explained by bronchiolitis under 1 year of age. There are major factors of deprivation and ethnicity, and ex-prems do much better than perhaps we give them credit for. Human metapneumoviruses and coronaviruses are out there, and we now have the ability to detect them. And they've been there a long time by the looks of things, at least 8,000 years for coronaviruses, possibly a lot less for human metapneumovirus. International cooperation goes without saying, really. It's been a feature, I think it's been a feature of the coronavirus pandemic, but I think going forward, that's going to be something that we're going to have to collaborate more closely with.